Good evening, Dr. Beau. It's a pleasure to have you on Battling and Beating Cancer.
Thank you, Scott. It's a pleasure to be here.
I have to say that we are so excited about the partnership that the Chicago Blood Cancer Foundation has with the University of Chicago and the other institutions in this area, because I think in addition to the tremendous work that you and your colleagues are doing here, to have the partnership of individuals like me who lack no talent, who can't do any research in the labs, but can bring some awareness and help generate some money so that you folks can do the magic that you do and that we're going to talk about later today. It's terrific to be able to partner with you. I wanted to start out and just talk a little bit about, you obviously could have done anything from modeling to rocket science, and you went into medical research. And can you just give our listeners a little bit of an idea of what it was about that that attracted you into medical research?
Well, Scott, I trained as a geneticist, and I didn't expect to pursue a career in cancer research. I came to the University of Chicago to do my postdoctoral fellowship, and I had the great good fortune of being mentored by Dr. Janet Rowley, who is one of the real pioneers in leukemia research. And within 3 months, I realized that I would never leave cancer research, because understanding the genetic basis of cancer is like figuring out a huge puzzle. And it was extremely stimulating. And it continues to be 30 years later.
And I have to say, we're sort of in, to me, the golden ages of research, particularly with the blood cancers. The things that have been done in the last several years, the things that you're working on now. It's really exciting times and reason for everybody battling blood cancer, regardless of what their current prognosis may be, sort of a candle of hope to say hang in there, because there are more treatments that are going to be produced from the research that's going on.
Well, I couldn't agree with you more. The pace of biomedical research is mind-boggling. It is moving at so log-fold greater than it did 10 and 20 years ago. And the hardest part is having the computational approaches to keep up with the high-throughput approaches we're using in the laboratory. But you're absolutely right. I think the outcome of this work is going to be more targeted, less toxic therapies for cancer. And I hope we can talk about that in the next few minutes.
Absolutely. And let's start, though, by talking about the Comprehensive Cancer Center and the research that you guys are doing. And just tell us a little bit about University of Chicago's mission.
Well, our Comprehensive Cancer Center is the university's infrastructure to coordinate, strategic plan, and advance our cancer research. And we have over 200 members who are focusing on understanding the biological basis, developing better treatments, and developing ways to prevent cancer. And of course, that involves extensive engagement with our community. And
a lot of interdisciplinary approach to be able to draw on the talent of people of different disciplines, which University of Chicago does so well.
Well, science is different now than it was 20 and 30 years ago, and it really is team science now. And it is interdisciplinary, as you said. The greatest advances have come from bringing individuals in disparate fields together, and it becomes really transdisciplinary research. It becomes something that is greater than the sum of the parts.
And what I've been finding as I get more and more knowledgeable about this is the leaders in research, they're visionaries. And you guys not only look for the talents within your organization, but you step outside of the proprietary box and are willing to partner with other institutions who have capabilities, which is one of the things that the Chicago Blood Cancer Foundation is excited about fostering. Could you just talk about that a little bit?
Well, our institution has extensive interactions with other investigators at the other major medical centers in Chicago. And this is the way science is done. We need to share information. We need to have computerized databases that each investigator has access to at the different institutions. And that's the way you maximize the outcome of your work.
And let's start for people like me who are educationally challenged. Why is cancer research so important?
How much time do you have?
Well, let's do about 2 minutes on this.
All right, let's try and fit it in there. So cancer is rapidly approaching and will soon surpass heart disease as the leading cause of death in this nation. And the impact of cancer on our nation, the cost of cancer care is staggering. It's over $220 billion a year. And that does not, and the cost for loss of lives, years lived, quality of life, and productivity is inestimable. If you look at the advances in cancer research that we've made, 30 years ago, cancer was not detected at an early stage. We only detected large tumors. We had the therapies that were available were very empirical. They usually involved long hospital stays and were often painful, and they have resulted in short survival. What's happened in the last 30 years is that the annual cancer death rates have fallen every year since 2003. We now have better methods of detecting cancer, and so early screening is more effective. We treat cancers earlier. We treat them with less toxic, more personalized, or minimally invasive therapies. And the survival rates have gone up. We now have 14 million survivors in the US, and that's the power of biomedical research. I think most Americans would be very surprised to know that if you added up all of the dollars that were spent, the average American over 30 years has spent a total of $260 in cancer research. That's $8.60 a year. And I think if you ask them if they feel they've benefited from that, I think they would be more than willing to invest more than that sum.
Yeah, and it's a paltry sum when you consider that 1 out of 2 men in America, more than 1 out of 3 women in America during their lifetime, going to hear probably the most scary words they'll ever hear, that you have cancer.
That's correct.
And
that's correct.
So it's the economics and it's also the humanomics of cancer.
Yes.
And
that's
that
a
really
great term.
is one of the things that we talk about in battling and beating cancer, and that the Chicago Blood Cancer Foundation believes, is that blood cancer research is really the superhighway to curing cancer and also other diseases as well. From the standpoint of the role of the immune system in disease, from the standpoint of all the breakthroughs that have originated with blood cancer research. And I'm wondering if you could just talk about that, some of the breakthroughs in lymphoma and leukemia research and how they've benefited people with other conditions.
Well, the history of cancer research will back up what you've just said. Leukemia and lymphoma have really been the model diseases for the major breakthroughs in cancer research over the last 30 or 40 years. One simple reason is that it's always been very easy to access these tumor tissues. You can obtain a sample of the leukemia cells by just sampling an individual's blood, and that's a non-invasive method, an easy way to get the cells. But leukemia and lymphoma research, if you look at the milestones, the first chemotherapy was developed for leukemia and lymphoma. The first bone marrow transplants were developed. The first studies to define the genetic basis of cancer and our ultimate recognition that cancer was a genetic disease were based on studies done with leukemia. And ultimately, one of the first targeted therapies has been developed in leukemia. So most of what we've learned in leukemia has subsequently been transferred to other cancers, other solid tumors.
And what I tell a lot of people is, obviously, if you're impacted or your family's impacted by a particular type of cancer, obviously you want to support that. And we also know that there's a great deal of synergy. Work on one area helps another area. But if you don't have a particular enemy in a particular form of cancer and you wanted to have the biggest bang, history tells us that blood cancer research gives us the biggest bang for our research dollar. Is that fair?
That's fair to say.
We're going to talk about research, and I'm in awe as I walk the halls of the University of Chicago because of meeting people like you and Janet Raleigh, who we met on the elevator on the way up here, and it's sort of, in some ways, going through the halls of leukemia and lymphoma research. But let's just talk about some of the basics of leukemia. I don't like the way it sounds. I don't like the way you spell it. But could you just tell our audience a little bit about what leukemia is, what the types of leukemia are?
Leukemia is a cancer of the blood-forming cells. So all of our audience, I think, will be aware that there are many different types of cells that circulate in our body. And those cells carry oxygen, the red blood cells, they fight disease. And leukemia arises in the bone marrow where these blood cells are formed, and it essentially results in an arrest of maturation where some of these cells fail to mature and differentiate into the mature cells, and at the same time they also continue to proliferate. And what happens is that those proliferating cells are unchecked or unregulated, and they eventually take over and kind of crowd out, if you will, the normal cells. So patients often present with fatigue. They're often may have fevers, easy bruising, excessive bleeding if they accidentally cut themselves, and those are all manifestations of the failure of your normal blood cells to work properly because they've been crowded out by the leukemia cells. And we divide leukemia into two major forms, acute leukemia and chronic leukemia. Acute leukemia is a very aggressive disease, as the name suggests, and needs to be treated immediately. Chronic leukemia is I won't say benign disease, but it is a less aggressive disease.
More of something that is chronic and something people can live with for a period of years and years and decades.
Particularly with the current forms of treatment.
Yeah. In terms of, and one of the things that leukemia not only is a primary cancer, but I was one of the side effects for my chemos I was taking for lymphoma was a very aggressive form of leukemia.
So we mentioned earlier that we are thrilled that we have 14 million cancer survivors, and that's the benefit of more individuals surviving cancer. But one of the unexpected side effects of the therapies that we used for cancer is the development of second cancers in individuals who have been successfully treated. And there are several different types of second cancers, but one of the more aggressive forms is a disease called therapy-related acute myeloid leukemia. And that's a myeloid leukemia that arises in patients who are treated for cancer with chemotherapy or radiotherapy. It develops on average about 5 years after treatment. So this is a very cruel irony that an individual has gone through, successfully been treated for one cancer, and goes on to develop a second cancer. And this disease is unfortunately very refractory to therapy, and most of our patients don't survive more than a year. And so we have been very interested in this disease at the University of Chicago, and we have been involved in characterizing the genetic basis for this disease, understanding who is at risk, who is susceptible to develop this disease. And the ultimate goal, of course, is to use that information to treat individuals, to predict in advance of treatment of the primary malignancy, and to treat them appropriately to lower their risk of developing this disease.
Can you just describe for us a little bit the conventional or the standard treatments for people with leukemia and in general, the prognosis for the various forms?
Well, the standard therapies are chemotherapy regimens, and the patients typically receive a combination of drugs. And the initial forms of therapy are remission induction therapies, so that's several cycles of multiple combinations of drugs. And some patients will then be retreated with a consolidation therapy, which is kind of an intensification of the therapy several months after their initial therapy to really eradicate any remaining leukemia cells. And another form of therapy that is appropriate in some patients and has had a great effect on our survival rates is bone marrow transplantation. And that can be, patients can receive a transplant from a relative, a sibling, for example, or even now an unrelated donor who is matched with some of the immune markers. And the survivals for leukemia have gone up dramatically. You know, again, this is the benefit of biomedical research. In the 50s, almost no one survived leukemia. In the 60s, the overall survival rate was up to 14%, 15%. By 1970, mid-70s, it was about 35%, and now it's about 65%.
And, you know, really phenomenal when you consider, you know, not only the dramatic difference in the numbers, but the impact on people who are going through it, who have now every reason to be hopeful. Give us your perspective of the next 5, 10, 20 years in terms of where you see research going and how you see that impacting patients and survival rates.
So we mentioned earlier that biomedical research is moving at an incredible pace. And this is partly because of the high throughput approaches that we can apply now to study an individual's malignant cells. So the goal now is to profile an individual's genetic makeup. By this I mean their non-malignant tissues. And the reason for this is that our genetic makeup can affect how we respond to drugs. It affects the toxicity and the efficacy of drugs, how we metabolize drugs. So we have to know first how an individual's own genetic makeup is going to affect how they respond to their cancer drugs. Then we need to know the genetic profile of their tumor tissue. And we need to know that at multiple levels. We need to understand the cellular pathways that are deregulated as a result of the genetic mutations that cause that tumor. And we need to use that information to pick out the Achilles heel of that tumor and to target our therapies to hit those particular pathways. And we integrate the information from the two types of profiling. We will be able to develop more personalized medicine, personalized therapy for each patient based on that profile. We believe that these therapies will be less toxic, more specific, and that many of the cancers that we treat now will really become a chronic disease, you know, a disease that you live with rather than a disease that you die from.
So we're seeing sort of a transformation from, I think in one level, particularly with non-Hodgkin's lymphomas, for example, in the dozen years I've been dealing with it, I think the classifications have changed from about 3 dozen types to more than 60.
Yes.
And as you're able to understand the molecular biology, you can understand the differences in the disease, that these actually are different diseases in some ways, and begin to understand, well, how do we treat them? And I envision that in time we're going to go from, well, you have a certain type of cancer, so let's give you this particular regimen. We'll give you CHOP or CHOP-R or whatever the regimen may be, to let's hone in on your particular cancer cells and let's give you the specific drugs that do the best, most efficacious job of destroying them and leaving the rest of you alone.
That is exactly the goal.
And everybody hears chemotherapy, and when I heard I was going to have chemotherapy, that scared me more than the surgery, because you expect to get sick, and in my case, I was sick. But today, with Rituxan, the monoclonal antibodies, the smart chemos, and some doctors don't even like to call them chemos because of what they connote, but it just means chemical therapy. So if we have an aspirin, that's chemotherapy in a sense. But these smart drugs latch onto the proteins and don't kill them, but they trigger an immune response. And so what it seems to me researchers are doing is multiple. Not only you're looking for the cause of disease, you're looking for the specific makeup of the disease, the treatments for the disease, and at the same time also, so many patients over the years got sick or died from the treatments. And nowadays, we're coming up with less toxic treatments and also different things to help out. You can take a particular shot to boost your red count, your white count, your blue count, if you have a blue count. So it's really exciting. And I just wanted to talk a little bit about, for people like me, I call us the untalented people, that I could put on a white coat like you have over there and still not do anything in the lab. But what is the importance?
You could play tennis, actually.
I probably could, maybe not very well, but I'd give it a shot. But let's say, what is the role that private funding has? We know everybody goes after the government money. But what about the private fundraising? Because we have a lot of people out there who are invited or asked to donate, whether it's dinners, walks, runs, bike rides, but how important is the private funding?
So private funding really enables the most innovative breakthrough research, and let me explain what I mean by that. Our federal funding systems require an investigator to submit extensive preliminary data. And how do they get that preliminary data? That data is generated by seed funding that comes from philanthropy. So when you donate $1 to cancer research, that is so much more than 100 pennies, because what we do is to leverage that dollar to obtain many, many more dollars from the federal government or other agencies based on the preliminary work that we do.
And it's also that seed money, as you call it, that really drives the research, because once you get to the point where you're looking for federal dollars, you've got to, as you say, have things established. And
That's
so
right.
when federal funding is limited, as it has been in more recent years, the incentive is to go with the tried and true
That's
methods.
correct. Yes.
And what we really need are those cutting edge.
Yes, and that is exactly, you know, the catch-22 of some of our federal funding. We describe a project and we need to do that project. It does not allow us to take the research in some of the most exciting directions. So some of the highest risk, highest value research is not funded by federal funds, and it has to be supported by private funding, by philanthropy.
And that's where we all come in, and that's where I think it's incumbent upon all of us as survivors or people who are impacted by cancer to join the team of curing cancer by contributing to our scientists. Now, I was, years ago, we founded an event called the Lymphomathon, a 5K walk and run. And I never met Dr. Altman, but I met his wife, his widow.
Yes.
And she came, and she actually came with a check and made a donation. And she's a lovely lady.
Oh, she still is?
And I haven't seen her probably in 3 or 4 years. And she talked about her husband and his commitment. And it was just one of those stories that, you know, I'll never forget. But can you tell our audience a little bit about Dr. Altman and his work?
Well, Dr. Altman was really a pioneer in lymphoma research. He was a true visionary. We all owe him a debt of gratitude at the university because, of course, he was the founder of our cancer center and led us for, I think, over 20 years. But Dr. Altman took a different approach to lymphoma treatment. He took a really multidisciplinary approach, and he also emphasized the necessity and basis for staging lymphomas and characterizing what subtype of lymphoma the patient had. And he used that information to develop better therapies. And so the therapies that Dr. Altman developed reduced the death rates from lymphoma by 60% in the 1970s and 80s. So he was a true pioneer.
Now, we could go on for an hour talking about some of the visionaries and the pioneers in your institution, but there's a couple I can't leave here without talking about. One is you, and then one is your mentor, Janet Rowley, who is just phenomenal and recently was given the Presidential Medal of Freedom, which is, I think, the highest award that any civilian can get. And of course, you received a year or two ago from the American Cancer Society their highest award. Tell us about you, Dr. Rowley, and your work.
Well, Janet Rowley, not to be repetitive, but another pioneer in cancer research. Janet realized and predicted many years ago that cancer was a genetic disease, and she sought ways to study the genetic pattern of tumor cells. And she focused on the chromosomes themselves. She was the first person to define a chromosome translocation, a recurring translocation that actually identified or diagnosed or was a sine qua non of certain forms of leukemia. And she then went on after that to define many different genetic subtypes of leukemia and lymphoma. And it is her work that forms our understanding of the genetic basis of tumors and the targeted therapies of tumors. So let me explain. She defined the translocation involving chromosomes 9 and 22 in chronic myelogenous leukemia. And that work then led ultimately to the development of Gleevec, which has really been a miracle drug, if you will. It's the paradigm of targeted therapies. It's an oral medication, so you can take a pill form, and it has reduced the death rate from CML. We don't even know the total survival rate from these patients yet, because they haven't even reached the median survival. It's now over 7 years since the first group of patients have been treated with Gleevec, and it's about 95% survival, and it's out over 7 years.
That's phenomenal.
So we don't even know how many. It could be, decades, and it's a single drug.
And the thing that strikes me, we look at you, you're beautiful and elegant. You see pictures of Dr. Raleigh getting the Presidential Medal of Freedom, but it's not a matter of getting in front of a camera or magically doing things. It's a lot of hard work, a lot of hours in the lab, a lot of fits and starts and false starts and changing things, and a lot of curiosity.
Yes, yes.
What does it take to be a good researcher?
Well, there's an anonymous quote that research is characterized by an undiminished enthusiasm in the face of failure, and that's the way research is. I think of it as a continuum. You continue to make small steps, and as you said, it's incremental and it requires enormous dedication. For my part, it was endless days, not nights, but endless days in front of a microscope, looking at chromosome patterns in leukemia cells and defining new genetic changes in leukemia cells. Science is an enormously gratifying career. It can be frustrating at times because things don't always go the way you wish they would, but it's really a privileged profession because of the opportunity to make discoveries, the opportunity to advance clinical care for our patients, and to enhance human lives.
Now, this is off the menu of questions that I put together, but in listening to one of our missions at the Chicago Blood Cancer Foundation, it's not only the research, which is so critically important, raising money for research, but it's also to make sure that in the Chicagoland area, we continue to have the best clinicians, the best researchers, so that patients of today and tomorrow get the best care. What is it that we can do to make sure that the Chicagoland area continues to get the best and the brightest going into medicine and research?
Well, we have enormous intellectual capital in leukemia and lymphoma research and cancer research in the Chicago area. It is important for us to be able to support junior scientists in the laboratory through seed funding, through career development awards for young trainees, for graduate students, medical students, to be able to develop programs to allow graduate students to learn about medicine and medical students to learn about research, to develop this translational pipeline to train the next generation. And I think that our academic medical centers and universities in Chicago are among the best equipped to do this, to lead this effort.
And the thing that always amazes me is that the great doctors that we do have, and we've had the privilege of meeting over the years, are the ones that have always come out in the different organizations and events that we've done, whether it's to speak, to educate, whether it's to raise money, whether it's just to be there. And just so proud of the great people that we have in medicine and science in the Chicagoland area in general, and the University of Chicago. And let me just, in the last minute or two, give you an opportunity to sort of tell folks what you think is on the horizon in terms of research, and what you think the future holds for people who are battling cancer because of the work being done today in the labs.
Well, I think that science is being run by team science. It's best advanced now by investigators who bring a variety of approaches to study a tumor. We want to see the entire picture of the tumor. And some of the technologies that we have available now, the high throughput approaches, DNA sequencing, et cetera, allow us to develop an integrated picture of a tumor. The computational approaches to analyze that massive amount of data that we can generate by sequencing to develop this profile. We need to understand all of the cellular pathways that are disrupted in the tumor, how the cell relates to the tumor itself, how the tumor itself relates to the organ, the individual, and how the environment affects that. And what we will be developing is a comprehensive picture. And it's that information that will allow us to develop the personalized therapy for each patient.
And my final question of today, although I would love to do this for another hour or two, is as a researcher, what message would you give to somebody who's suffering through blood cancer, who's a patient right now being seen in a clinic?
Well, I think that all cancer patients need to have hope. They need to be secure that they are receiving the best care, that there are good treatments available. I think it's important for people to be proactive in their care and to be educated about leukemia, and there are a number of organizations like yours that are extremely important in educating our patients about their treatments. So I think that patients need to be secure in the future of leukemia research and treatment for this disease.
And it's so critical because years ago, if you bought yourself another year or 2 or 3 or 4, which of course has its own worth, you were buying yourself another year or 2 or 3 or 4. But I think now, when you're buying yourself another year or 2 or 3 or 4 in remission, or in a disease that you can contain, you're buying yourself more because you're buying yourself the next treatment and ultimately the cure. And Dr. Beau, it's because of people like you that all of us have hope.
Well, it's a privilege and a pleasure to be a cancer researcher, and such a pleasure to talk with you.
Thank you. Dr. Michelle Beau, this has been Battling and Beating Cancer.